right. Hello also from me, and thank you very much for this opportunity to participate in the Festival of Genomics. There are not many festivals going on around the globe right now, so even a festival in genomics sounds very exciting to participate. It is also a great opportunity for me to be in the office and escape from my children. So all in all, I'm very happy to be here. My name is Yanni Panagiotou, and I'm working on microbiome systems biology. I'm the head of systems biology at the Leibniz Institute for Natural Product Research, and also an associate professor at the Faculty of Medicine at the University of Hong Kong. My talk today is of course on microbiome. The whole session is on microbiome, and there is a reason for that. It's probably one of the hottest topics in both academic and industrial research, with a very high productivity from the scientists. And when I say high productivity, I really mean publications. If you look 10 years ago and you search in PubMed using the microbiome as the main keyword, you will get around 160 results, which means that if someone could basically read the whole literature in that year. But if you do the same in 2020, you will realize that there are many more papers published per year. It's actually around 23 papers that are published every day. So the ones that you are going to attend this session, you will be behind the literature by the end of the day. There will be papers that will be published while we are talking. So the question is, what have we learned from all these thousands of papers that have been published this last decade? And if I summarize it, I will say that we have learned three things. The first one is that, as also Florian mentioned in the previous talk, is that even though the two humans have a very high similarity in terms of their DNA, up to 99.9%, two unrelated humans share less than 10% of their microbiome. And this is something that we should remember, and I will come back to this when I will show you some case studies when we try to design microbiome-based therapeutics, these differences between individuals. So we have this microbial signature, and this microbial signature is actually influenced with whatever we do. Every lifestyle choice that we make is going to change or have an impact on our microbiome. And one such example, a couple of years ago, we published a paper where we could show that if we take a sample from your skin microbiome, we can actually find out which metro line you have been using in a mega city like Hong Kong. So there are 10 different metro lines, and if we do shotgun metagenomics on your skin, we can say which metro line you used and if you use it in the morning or in the afternoon. Of course, there's not a big clinical value on this. You can basically ask someone which metro line he used. You don't need to sequence the skin microbiome. But this probably was one of the things that also helped teams find new applications for the microbiome in forensics. There are many teams that have been working to use the microbiome in forensics, including the team of Horatio in CSI Miami, that he used actually the microbiome in one of the episodes to solve a murder case. So of course, if Horatio is using it, it must be true, right? The second thing that we have learned is that there is an association between many diseases and the human gut. How new is this? Not really. I mean, it's the Greek doctor Hippocrates that actually had this famous phrase that all diseases begin in the gut around 2,500 years ago. How people reacted back then about this statement, we don't know. There are always people in denial. But now with the next generation sequencing, we know that even if it's not all diseases that they begin in the gut, there are many diseases that are actually associated with changes in the gut microbiome. And usually the framework that we apply for making these associations is that we take cohorts that they are either healthy or they have a disease. We take a stool sample, we do metagenomic sequencing, we characterize both the taxonomy and the functionality of the microbiome, and then we use machine learning tools in order to see if we can classify based on the composition the healthy and the diseased people. But besides that, I think what really revolutionized the field is that we can actually prove also causality. So we have mice models, either germ-free mice or gnotobiotic mice, and we can actually prove that the drivers of this differentiation between healthy and disease are actually causing the disease, and they are not the result of the disease. So what is next? I think the third and probably the most interesting thing that we have learned the last 10 years is that we can actually rationally edit the microbiome. This is the most interesting part because we realize that you can change the human phenotype by modifying the microbiome. And there are four, I would say, main ways to change the microbiome composition. Already 6 years ago, the team of Peter Turnbaum published this paper in Nature about the role of diet. He did some very elegant interventions where participants were either given a plant-based diet or a meat-based diet, and he could see very clear impact on the composition. But besides diet, there are other ways to modify the microbiome composition: exercise, supplements. And supplements could be probiotics, could be prebiotics, symbiotics, you name it. And the last tool in our ability to change the microbiome composition is with CRISPR methods, where you can selectively kill bacteria. And there is a Danish company, Sniper, with this very cool name that is actually doing that very well. So what I will do in the next slide, I will give you some examples from my group, from my lab, using these methods, diet, exercise, and supplements to actually modify the host's phenotype. And I will start with probiotics. So cancer, the association of microbes with cancer is also not new. We know, for example, that for years that Helicobacter pylori is a risk factor for developing gastric cancer. But we didn't know much about the role of the gut microbiome for extraintestinal cancers. So we looked into this a couple of years ago. We designed a novel probiotic mixture, and this design, actually, the process of designing it didn't make it to the final paper because it was a lot of work to read a lot about the literature and do a lot of preliminary experiments to figure out what could be an interesting cocktail of bacteria. So we ended up with this mixture of around 15 to 20 bacterial species. And then we used mice to test the impact of this probiotic mixture in hepatocellular carcinoma. We had four different mice groups. In some of them, we gave the mixture, this ProHep, either at the same time when we did the tumor injection or a week before, in order to prepare the immune system. And then we had the group of mice that served as controls treated with cisplatin, which is an anti-cancer drug, or just with normal diet. And what you can see on the figure on the right is the impact that the probiotic mixture has in the tumor size. And actually, the probiotic mixture given 1 week before the tumor injection could reduce the tumor size up to 40%. So that was really a very interesting result that we wanted to find out a little bit more how this can be possible. So there's a lot of mechanistic analysis in this paper, but I will only focus on the T cell differentiation. So we could find out that the tumor size and the tumor volume in this type of cancer was very much correlated with the frequency of the Th17 cells in the tumor. And then when we analyze different tissues in the mice, we realized that the impact that the probiotics have is actually by reducing the levels of Th17 cells in the intestine and in the blood. So basically, they are inhibiting the process of recruiting Th17 cells through the blood circulation from the gut to the tumor. And then, in order to bring more the microbiome composition into this process, we did shotgun metagenomics, and we could link specific bacteria species that produce anti-inflammatory metabolites and regulate this T cell differentiation as the way that probiotics have a positive impact in reducing the cancer progression. Then, of course, we started looking into humans, but there was already a great competition. Other groups had also started looking on how the microbiome can influence anti-cancer treatment. So there was 2 great papers published in 2018 where they show very clearly that whether you will respond or not to immunotherapy depends really on the composition of your gut microbiome. And they look into 2 groups of patients, on melanoma patients, and then on lung and kidney tumors in order to prove this theory. As I said, at the same time, we were also looking into this, and we actually added a little bit on this knowledge. We had a cohort with many different cancers, and actually treated some of the patients treated with immunotherapy or a combination of immunotherapy and chemotherapy, and we could actually show that there are specific species that they can have a positive or a negative impact on the treatment, independently the cancer type and independently the type of therapy, that they have really a strong influence on the immune response. And actually, we could also develop a machine learning model using the baseline microbiome. That means the microbiome before we initiate any therapy, and see if we can predict who is going to respond and who is not going to respond to therapy. And it was a very high classification, up to 90% in this cohort. And then we tested also in another small cohort, which also worked pretty well. The second way to modify the microbiome is with exercise. And of course, exercise has been recommended for many different metabolic diseases, including diabetes. The problem with diabetes is that it's a lifelong disease. You cannot really reverse it, but for pre-diabetes, it's a reversible state. So if you go to your doctor after you have been diagnosed with pre-diabetes, what he will probably tell you is that you need to exercise more. What he's not going to tell you is that around 7 to 65% do not respond to exercise, or they actually respond negatively to exercise in terms of insulin sensitivity and glucose levels in their blood. So we started thinking that whether again the gut microbiome could be a way, could be the mediator of the beneficial impact of exercise. And since there are no prognostic factors whether someone is going to respond or not to exercise, if we can use the gut microbiome as a way to predict who is going to benefit from an exercise intervention. So we designed this clinical study, this training intervention. So we had a group of people, of 20 obese or overweight Chinese individuals, that they entered this high intensity training. And we had also 20 lucky ones that they were into the lazy group, that they didn't have to change anything in terms of exercise. And of course, we monitor them very closely for 12 weeks, and we also sequenced their gut microbiome. And if you see the response of these 20 people in terms of the HOMA-IR index and the fasting insulin, what you will see is exactly what you were expecting based on the literature. So from these 20 individuals, 14 responded and 6 did not respond. So in this figure, the red lines that we follow them longitudinally are the non-responders, where you can see that both the HOMA-IR and the fasting insulin did not improve, but the other 14, they had a clear improvement in their metabolic state. And it was, of course, obvious that we will try to see and prove that this improvement is because of the changes in the gut microbiome. So we did fecal transplantation. We used feces from responders after the exercise, and feces from the non-responders again after the exercise. And we could see that the improvement in the metabolic parameters in mice were only observed with the responders. And to make sure that this was not something that already existed there before the exercise, we also performed the fecal transplantation with a baseline fecal samples, and we could see that there is no difference. So clearly, the microbiome here plays a major role in improving your glucose levels after exercise. And then the second question that we had already posed from the beginning is if we can use the baseline microbiome as a predictor of whether someone is going to benefit from exercise or not. And indeed, we could again build a random forest model and predict with an accuracy of almost 90% if exercise is for you or not. And then we used also a validation cohort in another country. So we ran it, the discovery cohort was in Hong Kong, then in another clinic in mainland China. We did the same, and again the accuracy was up to 80%. So I think this could be a very valuable tool for clinicians. We need to do it, of course, in a much larger cohort, but it seems very, very promising. And of course, then there was a lot of work to understand the mechanisms exactly. And this is just a graphical abstract that shows that what exercise is doing into the microbiome is really changing the ability to produce short-chain fatty acids and decrease the production of detrimental compounds like amino acids, branched-chain amino acids, and other compounds. So in this case, because you will see in my next case study, in this case we could not pinpoint one specific bacteria species that can be used therapeutically, but it's a whole community that is changing in a positive direction. So the last case study that I will present you today from my group is about obesity, and I think obesity was probably the first phenotype that was clearly linked with dysbiosis in the gut. And I think these two maps here helped a lot the researchers understand this association between the gut microbiome and obesity. So the map on the left is showing you how the prevalence of obesity is changing the last 30 years in US, and you can see that from almost a non-existing problem 30 years ago, it has become really a pandemic. And then on the right, you can see a map. Unfortunately, I couldn't find an interactive map like the other one that show you the whole trajectory of the last 30 years, but this is a snapshot of the 2010. But I think you can see very, very big similarities between these two maps, antibiotic users and obesity. And we know, and you heard also from Florian in the previous talk, that antibiotics have as a side effect, as an off-target, the gut bacteria. So this was a clear evidence that we have to look into the microbiome if we want to understand why we have this obesity epidemic. And another famous thing that Hippocrates has said is that we are what we eat, and food could be our medicine. The thing is that as a Greek, I have heard the same thing from my mother for so long that I thought that my mother was the first one that actually said, "Let food be the medicine." But then I figured out that there was also Hippocrates that have said the same. So we decided to look into dietary interventions, but not dietary interventions that they target the host, but dietary interventions that target the gut microbiome. So we decided to make a clinical intervention with resistant starch. Resistant starch is a type of polysaccharide that cannot be absorbed by humans, but it goes directly to the gut for the bacteria, so it's food for the bacteria. And we had also a control group with control starch, but this is a crossover design, so we had both groups going on the same two interventions with a washout period. And you can see in the two figures at the bottom that really the interventions, the people that were eating this resistant starch had a significant improvement in the body weight and also in the fat mass. And this is just two of the parameters that I decided to show you, but everything that we measure related to lipid absorption was really improved in this group. And then, if you look into the changes of the microbiome composition of these two interventions, you can clearly see that resistant starch, it brings the community to a completely different structure than the one that the control starch. So this is an NMDS plot based on the fold change of different bacterial species. And then if you try to correlate the most important changes at the bacterial species level with the most relevant parameters like the BMI or the visceral fat area or the waist circumference, you will see that there was always one specific bacterium, Bifidobacterium adelocentis, that was strongly suggested as a driver of these changes. So in this case, besides of course the fecal transplantation that we did with the feces from people that weren't they had a diet of resistant starch and the diets with control starch, and we could of course replicate the same findings as in humans. We also decided to look into one specific species. So we did monocolonization of mice, obese mice, with Bifidobacterium adolescentis and also with heat kill, and we could see that we can replicate the positive impact only with the live Bifidobacterium. So it is something that it is produced and it's needed in order to see this important host improvement. So this is in contrast to what I told you about the exercise, that we couldn't really identify one species to be responsible for the phenotype. A lot of experiments, and there's a lot of this is not published yet. We hope that it will be published soon. We could show that just one bacterium species can be used in the clinic for improving, and mechanistically the major impact is through this AGL PTL4 protein, which is regulating the lipid absorption. So with that, I think I'm reaching the end. What I often been asked is what I see then next in microbiome. I think many of the groups, including our group, we are working on long-term prediction of disease development. So how can whether you can use the microbiome to predict a disease that is going to be developed in 3 or 4 years. We just finished a paper on predicting non-alcoholic fatty liver disease using stool samples 4 years before, and it's very promising. We can predict with an accuracy of around 77%. So I think that's already a sign that in combination maybe with other clinical parameters, we can develop these long-term predictive tools. Design prophylactic approaches, and also this microbiome navigation system that is suggested by Rob Knight, that I just think it should be continent specific, and there will be probably a different one for Chinese people, different for Europeans, and the MGPS that Rob Knight is developing for US. I think that I will only say some acknowledgements, because then I will not have time for any questions if there are questions. So I would like, of course, to thank Novogen, who have been working very closely together the last years, because for many of our projects we needed Novogen to develop specialized methodologies. I didn't present here about the tissue microbiome, but this is a big thing in our lab, and Novogen has set up the pipelines for analyzing the tissue microbiome. We have a lot of projects where we are looking on fungi, and this needed also customized protocols. So really a big thank you to Novogen for this. And of course the funding bodies, my group, and the main people that have been in these case studies that I presented. And thank you very much for your time. Yes, are we able to recommend specific dietary interventions to optimize patient microbiomes in pre-diabetes to alter the non-responder microbiome to be more like responder microbiome? I think we can. There are so many now, all these association studies that have been criticized because they were not proving any causality. I think they can be used now to link with these microbiome source algorithms the disease phenotype with dietary. We have tested a lot of different things now, and even the impact of drugs. So I think we can start designing based on the disease microbiome specific interventions. Could be dietary, could be drugs, could be anything. But now it's the time of integrating all this data that we have. And with that, I think I have to stop. Thank you very much.